Hi everyone, welcome to topic 3, chromosomes. Um, all right, so first let's take a look at a karyotype of the human genome. So a karyotype is essentially a picture of the chromosomes within an organism. So in different species, our karyotypes are going to look different because different species have different number of chromosomes. For our purposes, we're mostly going to look at the human genome or karyotypes of humans. And so a normal karyotype for humans is going to have 22 autosomal chromosomes and 22 pairs of autosomal chromosomes. These are non-sex chromosomes. And we're going to have one pair of sex chromosomes. So human genome contains how many pairs of chromosomes? How many pairs of those are autosomal chromosomes? And how many pairs are sex chromosomes? All right, excellent. So you should have gotten 23 pairs of chromosomes, 22 autosomal, and one sex chromosome, one pair of sex chromosomes. All right, and so the presence of a Y chromosome would make an individual biologically male. Okay, so and we'll look at some examples of that, of some abnormal karyotypes, trying to determine which biological sex the individual would be. But I do have a question for you now. Why do we have pairs of each chromosome? For example, let's take a look at chromosome 8. Why do we have a pair of chromosomes? Where do you think we get each of these chromosomes from? Right, so essentially, one chromosome is from the female parent, or parent A, and one chromosome is from parent B. So now let's take a look at a karyotype of someone who has Down syndrome. What do you notice is different about this karyotype? Excellent. So we have 3 chromosome number 13s. This is called a trisomy, when we have an extra copy of a chromosome present in the cell nuclei. Let's look at this example. Where do you think this third chromosome came from? Right, excellent. So 2 of these chromosomes came from parent A, and 1 copy was given by parent B. We can't determine which parent is the one that gave 2 copies of chromosome 13, but we know that one parent passed on 2 chromosome 13 chromosomes, while the other parent passed on 1. Why does this happen? This happens as a result of non-disjunction and meiosis, which we'll talk about next topic. All right, now take a moment, please, and look at this karyotype of an individual with Turner syndrome. What do you notice is abnormal about this karyotype? Okay, excellent. So our pairs of autosomal chromosomes all appear normal. Chromosomes 1 through 22 all have 2 copies, which is excellent. But if we look at the sex chromosomes, we notice an abnormality. We are missing the second chromosome, second sex chromosome, resulting in a monosomy when we have an absence of a chromosome. It's my question for you is, do you think that someone with Turner syndrome, this is their karyotype, do you believe that they would be biologically male or biologically female? And last but not least, let's take a look at a third karyotype of a person with Klinefelter syndrome. Please identify the mutation. Excellent. Okay, so our autosomal chromosomes are all normal. We have a pair of each autosomal chromosome, but if you notice, we have a trisomy here. We have 3 sex chromosomes, 2 X chromosomes, and 1 Y chromosome. What biological sex do you think this individual would be, and why? Okay, so now we're going to step away from the karyotypes, now that we understand what the human genome contains, the chromosomes we find in the human genome, and some abnormalities that we find within the human population. And let's move on to some key terms for our next topic. So for this slide, we're going to focus on traits and alleles. For example, a trait is a characteristic that an organism inherits, and then alleles are variations of that trait. So, for example, we have eye color. eye color would be our trait. And then the different options that we can inherit for eye color would be called alleles. So my trait would be eye color, and then brown eyes would be an allele, blue eyes would be an allele, green eyes would be an allele. Okay, and so now let's move on to another one. We have hair texture, we have curly hair. Out of hair texture and curly hair, which one is the trait and which one is the allele? Excellent. Hair texture would be our trait. And then what are the different options that we can inherit for this trait? We can inherit curly hair, we can inherit straight hair. So curly and straight hair would be our alleles, and hair texture would be our trait. All right, now let's look at height, short, and tall. Please identify the traits and alleles, and do the same thing for petal color. All right, so now that we understand some terminology, we can move on to Mendel's experiments. So Gregor Mendel grew up or was born and lived in what is now the Czech Republic in the 1800s. He was a biologist, a meteorologist, and a mathematician. So he really liked to study the way math is integrated in biology and meteorology. And his research started with a question, like all research. And he wanted to know, is there a pattern to inheritance? Can we predict the probability of an allele being inherited or expressed in the offspring of individuals? For example, if we have a tall parent and a short parent, what is the likelihood that their children would be tall or short or medium? Is there a way that we can use math to predict what characteristics the offspring would have for parental generation? And so, since he loved to be in the garden, he decided to do research using his pea plants. And he looked at various traits, such as tall and short, and he looked at the colors of the petals for the flowers on the pea plants. And he looked at the colors of the pea pods themselves, and to better understand inheritance and the mathematics behind inheritance. So, for example, one of his experiments, he would take tall pea plants, purebred tall pea plants, meaning that they were tall, their parents were tall, their grandparents were tall, their great-great-grandparents were tall. These tall pea plants have always been tall. Okay, they have nothing in their genome that would indicate that they have ever been short or that they will ever be short. And he decided to cross-pollinate them with pea plants that have always been short. So he took purebred tall and he crossed them with purebred short pea plants. And at that time, his prediction, his hypothesis was that the offspring would all be medium in height. They would somehow be a combination of the two. But what he found was 100% of the next generation was tall, which was kind of strange. It was like the short characteristic or the short allele just disappeared. So then he said, okay, well, what if I take these tall pea plants, the second generation here, and what if I cross them with one another? What would the resulting generation be, this second generation? And surprisingly, he found that the short allele came back. So 75% of the second generation maintained the tall characteristic, expressed the tall characteristic, the tall allele, and 25% were short. So from his experiments, what did he conclude? He concluded that, first of all, some alleles are dominant to others. Okay, so if we cross two individuals that are purebred for traits, there are some traits that are dominant or that will overpower or will be expressed over other alleles. And we determined, and that some alleles are recessive. They can be hidden in generations and then expressed later in later generations. So today we indicate that's where we describe these alleles as dominant and recessive. Our dominant alleles we represent with capital letters, and recessive alleles we represent with lowercase letters. So let's look at some examples. Freckles are dominant to no freckles. So which letter would represent freckles, the freckles allele, and which letter would represent the no freckles allele? Tall pea plants are dominant to short. So which letter would represent our tall pea plants, and which letter would represent our short pea plants? So actually, yes, S can represent the tall allele. A capital S can represent the tall allele. So we don't have to use T for tall or F for freckles. We can actually use any letter of the alphabet we'd like. Generally, the letters that are chosen match the name of the allele, but they don't have to. And usually letters that are chosen are letters that you can easily tell the difference between the capital and the lowercase. For example, T, the capital T and the lowercase t look distinctly different, so it's very easy to distinguish between them. So it's an often chose letter. Letters like P, which look very similar as a capital and lowercase, are less likely to be chosen. So let's take a closer look at what these chromosomes look like inside of a cell and what these alleles, look like. So for example, we have 23 pairs of chromosomes in a cell. So let's just look at one pair now. So this is one pair, and along this chromosome, there's the codes for different genes, okay, or different proteins. But for example, this region of a chromosome will match this region of the chromosome. So if this region codes for freckles, this one will also code for freckles. Okay, these are homologous chromosomes. So the location of the gene on one chromosome will match the other one. Okay, so for example, this is the gene locus for freckles, so the gene location. And let's imagine if someone is homozygous dominant for the trait, that means that they have two dominant alleles. So what would homozygous dominant look like? It would look like a dominant allele on both chromosomes. It would be homozygous dominant. Now, if we're going to look at homozygous recessive, if we say someone is homozygous recessive for freckles, that means that they have two recessive alleles on their chromosomes. And if we say that an individual is heterozygous, that means that they have one dominant and one recessive, and it can be either order. We don't know which chromosome necessarily is the dominant one or the recessive one. We just know that one of them is dominant, one of them is recessive. So let's imagine that an individual inherited the freckles allele from parent A and the no freckles allele from parent B. What would the genotype of this individual be? And what would the phenotype of them be? So phenotype is their physical appearance, and genotype would be, you can either say the letters like capital F, or you can say something like homozygous dominant. Either one of those terms would describe the genotype of an individual. Phenotype would be, do they have freckles or they don't have freckles? So for these answers, you should have said that the genotype for an individual that got a freckles and no freckles allele is heterozygous, okay, or big F, small f. And their phenotype, so they have a freckles allele, and freckles is dominant, so they will express the freckles trait. In order for us to express that recessive trait, that means that we need to have homozygous recessive. All right, so now a pea plant is heterozygous for height and homozygous recessive for flower color. So what would the plant alleles be on each chromosome? And what would the phenotype of the plant be? Let's take a look at this. For height, it's heterozygous. Try to answer. And for flower color, it is homozygous recessive. What would those genotypes be? And what would the phenotype of the plant be? So would it be tall? Would it be short? Would it be purple? Would it be white? Would it have purple petals or white petals? Here's some vocabulary terms if you would like to review them. And so now let's take a look at this in the context of a real disease, cystic fibrosis. Okay. So recall, the cystic fibrosis is found on chromosome 7. So we're imagining that these are homologous chromosome 7s. And the result is the deletion of the 508th triplet in the DNA code. So The result is a non-functional chloride transport protein. So this is a recessive characteristic, the non-functional chloride transport protein, and then the dominant would be the functional chloride transport protein. So why would a person heterozygous for cystic fibrosis not display the symptoms of the disorder? Essentially, what I'm asking, how do we know that the non-functional one is recessive and the functional one, why would the functional one be dominant? And so let's take a look at this for a second. This chromosome, if we were to use it to make a protein, we wouldn't be able to make the chloride transport protein, because this is our mutated one. And it results in a non-functional transport protein. Okay, so it doesn't give us the correct protein. However, this individual, this one, it has the ability to make a functional chloride protein with its other chromosome. So would this individual be harmed by that mutation? No, not really, because they still have the code to make a functional one. Right? So why is this dominant? Why is the functional one dominant? The dominant allele? Because at least we have it. We have the instructions, so we're able to make the functional protein, so it's not a big deal. If the only way that the mutated one would be the only way that an individual would be affected by the mutated one is if both were mutated. Okay? If both were mutated, then our individual would have no way to make the functional protein. So we need both to be mutated for them to not be able to make the protein at all. And that is why the mutated one, or the non-functional allele, is recessive. All right, so now let's look at the next passage. The Huntington's disease, is it dominant or recessive? And would someone heterozygous for Huntington's disease display symptoms of the disease if they're heterozygous? And why would they still display symptoms despite having one allele for a healthy protein? Yeah, so you should have got that the Huntington's disease is dominant. And if someone is heterozygous, that means because it's dominant, they would still have Huntington's disease. And why do they still have symptoms even if they have a healthy allele? So in this case, they have one healthy allele and they have one mutated one. But why do they still display the disease? Because just having the mutated protein will, in your having the code to make the extra long protein, essentially starts poisoning your body, right? The extra long protein harms the nerve cells. So if we're making that protein, then we're harming our nerve cells. So we have Huntington's disease. So it's a dominant trait because just having that mutated protein, that protein that's extra long, the abnormal protein, harms the nerve cells. Right? So even though we're making the healthy protein, the correct protein, because we're also making the abnormal protein, we're displaying the traits of Huntington's disease. Comparative to cystic fibrosis, which the mutated one's not harming us. Okay, it's just sitting there. The abnormal protein is just sitting there, right? And they're still able to make a functional one. So as long as they're able to make a functional one, everything's fine. Okay? So